Hello and welcome to Badger Talks Live, which brings exciting happenings, resources, and talent from your UW flagship campus to the people of Wisconsin and beyond. I am Megan Bakabuk, a third-year Ph.D. candidate studying cellular and molecular biology in the School of Medicine and Public Health. I'm originally from San Diego, California, and over the past couple of years, I have worked in the Wei Xu lab studying a novel target to combat endocrine-resistant breast cancer. During the month of September, Badger Talks Live is proud to showcase the latest cancer research happening at the McArdle Laboratory for Cancer Research. I'm pleased to introduce Dr. Wei Xu, a Marian A. Messerschmidt Professor of Oncology at UW-Madison. Today, she'll be sharing information about the history of breast cancer, the biology underlying its development, statistics about breast cancer in Wisconsin, and the latest personalized treatment options. Wei went to Peking University for her undergraduate studies in chemistry, then went on to receive her MS degree in biophysics from the Institute of Biophysics at the Chinese Academy, China. She obtained her Ph.D. in biochemistry from the University of Iowa and received her postdoctoral training at the Salk Institute for Biological Science. Her research addresses fundamental mechanisms of endocrine resistance and identifies the biological activities that go awry in disease states. By identifying novel targets in endocrine-resistant and triple-negative breast cancers, the Xu Lab develops new therapeutic strategies to control disease progression and prevent recurrence. Questions will be answered at the end of the presentation, but feel free to post them in the chat at any point. Please welcome Dr. Wei Xu.
Thanks, Megan, for the very nice introduction. So it is indeed my great pleasure to give the third Badger Talks in the month of September, highlighting research at the McArdle Laboratory for Cancer Research. So my talk will cover historical discovery and treatment, main challenge and treatment strategies. At the end, I will touch upon precision medicine and future perspective. So breast cancer is the most frequently diagnosed cancer in women in the majority of the country. So 1 in 8 women has a lifetime risk of developing breast cancer. So this is the anatomy of breast cancer. So the breast has 15 to 20 lobules. The cells in these lobules could secrete milk during breastfeeding. The milk are transduced through these ducts and going to the nipple. So at the back of this breast is the muscle. So it helps to attach the breast to the rib. So there are usually 30 to 50 lymph nodes surrounding the breast. Breast cancer arises from abnormal growth of cells in ducts and lobules. So here is a cross section of the ducts. If tumor cells is localized inside the ducts, they're called ductal carcinoma in situ. If the tumor cell breach out and get out of these ducts, they are called invasive ductal carcinoma. So breast cancer cell could also arise from these lobules. So if the cancer cells are localized in the lobules, they're called lobular carcinoma in situ. If they get out of this space, they are called invasive lobular carcinoma. Invasive ductal carcinoma accounts for the majority of the invasive breast cancer. So the breast cancer is the most frequently diagnosed cancer. The incidence in state of Wisconsin is very similar to the national trend. So at the incidence rate, breast cancer is the most frequently diagnosed cancer in Wisconsin women. And the second in the cancer-related deaths. So if we combine all the man and woman cancers, breast cancer is listed in the as the 5th position in causing cancer deaths. In the state of Wisconsin, it is estimated around 720 women will die from breast cancer in this year. So here are some like a distribution of breast cancer in the state of Wisconsin. the left panel shows the incidence rate by county in Wisconsin. The right shows the mortality. So you can see in the northern part of the northern counties, some counties have a very high incidence rate as well as the mortality rate as compared to the Dan County. So due to our understanding of breast cancer biology and the advancement of breast cancer treatment, the breast cancer mortality has declined since 1975. So today I will give some historical overview of breast cancer treatment, highlight some major discovery for breast cancer treatment, and discuss the biology underlying those treatments and what we are up now. So in 1882, William Hastad operated a breast surgery called radical mastectomy. So this procedure removed whole breast, all the lymph nodes under the arm and the chest wall muscle. So this surgery increased the breast cancer survival rate, 5-year survival rate up to 80%. So the radical mastectomy has been the standard of the surgery for many years. But because this procedure is very brutal and could cause deconfiguring of women, so this is rarely performed nowadays. So the British surgeon, George Peterson, so he found that if he performed the ovaryectomy, this is a procedure to remove both ovary from a woman, he could dramatically extend the life of women with metastatic breast cancer. So this provided the first hint that ovaries secrete a substance that could promote breast cancer progression. So in 1923, 2 American scientists purified estrogen from ovarian follicle of hog. So this is the structure of 17-beta estradiol. This is the major estrogen in women. So they show in animal, this hormone could increase uterine weight, stimulate vaginal maturation, and also increase sexual receptivity. So in women at reproductive age, 17-beta estradiol is also the major female hormone that promote the reproductive organ development, also maintain the sexual desire. So, so we know this hormone, so what this hormone binds to. So in 1958, Elwood Jensen, a biochemist, purified estrogen receptor from rat uterine tissue. So with the purified protein, he was able to generate an antibody. So when he sent the antibody to the pathology lab, it is clear about 70% of all breast cancer express estrogen receptor. So he also noticed that cancers expressing significant level of ER benefit from ovary removal and anti-estrogen therapy. So this fundamental work established ER as a predictive marker for endocrine therapy. So here are a few examples showing the ER staining in breast tissue. So on the left, this is a breast specimen that has a heavy ER staining. You can see that ER is strongly stained and almost all the cells express ER. So this middle sample show a weak estrogen receptor staining. So the right sample show a strong ER staining, but only a small subset of cells express ER. Most clinicians, they think that if the breast tumor, about 10%, over 10% of breast tumor cell express ER, they call ER positive tumor, and the patient are advised to be treated with endocrine therapy. So due to the declaration of a war on cancer by President Nixon, so Craig Jordan who was a faculty member in the Department of Human Oncology, tested if tamoxifen could be used for treatment of breast cancer. So tamoxifen at that time was synthesized by AstraZeneca as a contraceptive drug, but it was found to be not very effective. So it was about to be abandoned. So Craig Jordan showed the tamoxifen could inhibit tumor growth in rats. In addition, tamoxifen inhibits the initiation of carcinogen-induced rat mammary tumor. So this study led to approval of tamoxifen for treatment of ER-positive breast cancer. So the discovery of breast cancer prevention property of tamoxifen also leads to approval of tamoxifen for chemoprevention. So treatment of ER-positive breast cancer by blocking estrogen action are collectively called anti-estrogen therapy. So sometimes they are referred as hormone therapy or endocrine therapy. So Craig generously shared some pictures with me. So this is show the Wisconsin tamoxifen team in the 1980s. So you can see the senior leadership of UW-CCC pay a great deal of attention to the tamoxifen study. So sitting in the front is Howard Rush. So he is the founder of Mercado Laboratory. In 1973, he established the UW Comprehensive Cancer Center. Paul Carbone is the subsequent director of UW-CCC. So the Carbone Cancer Center is named after him. So data from a larger clinical trial show tamoxifen, a long-term use of tamoxifen. So these are data from 5 years, could dramatically reduce the recurrence and the mortality of breast cancer. It is estimated 500,000 women are alive because of tamoxifen. So the using tamoxifen for breast cancer treatment is one of the greatest advances in the medical oncology history. So the simple mechanism is tamoxifen block the action of estrogen. But we have to keep in mind though, tamoxifen is not always antagonizing estrogen's function. So in breast, tamoxifen antagonizes estrogen's effect. But in other tissues, tamoxifen could behave just like estrogen. So for example, long-term use of tamoxifen could also increase the woman's risk of developing endometrial cancer. So since we know the estrogen, the next question becomes how estrogen works and how does tamoxifen block the effect of estrogen. So the major breakthrough was made by Pure Champong when he cloned the estrogen receptor. He revealed the DNA sequence encoding the estrogen receptor we call ER. So once the DNA sequence is known, it's very clear to biologists. So this protein could exist in the nucleus to regulate gene expression. So the DBD stands for DNA binding domain. So that tells us this protein could be in the nucleus binding to the DNA. And the LBD stands for ligand binding domain. So this is where estrogen bind, environmental estrogen-like compound bind, and also phytoestrogen from plants bind. So Pure Champong also showed the estrogen receptor, when it's getting into the nucleus, it forms a homodimer binds to DNA. So around the same time, Dr. Ron Evans, my former postdoc mentor, cloned the glucocorticoid receptor. So together, their work laid a foundation for the discovery of a superfamily of nuclear hormone receptor that could sense a variety of hormones in our body. So Pure Champon, Ron Evans, together with Edward Jensen, who purified the estrogen receptor, they won the Albert Lasker Award in 2004. So this is an animation slide that show estrogen works and how tamoxifen block the effect of estrogen. This red circle stands for estrogen. This is estrogen receptor bind to estrogen. So when estrogen receptor bind to estrogen, it's a form a dimer that binds to the estrogen responsive element in DNA. So where it can recruit a lot of proteins to regulate gene expression. So those genes are typically involved in promoting growth of a tumor or have an anti-death signal or promote migration of the tumor cells. So when tamoxifen is present, tamoxifen compete with estrogen for the same binding site. So it's a prevent expression of those genes. Aromatase inhibitor, the hormone therapy agent given to the postmenopausal woman. Aromatase are the enzyme enriched in the fat surrounding the breast. So the function of this enzyme is to convert the male hormone testosterone to the female hormone 17-beta-estradiol. That's increasing estrogen concentration in the breast. Aromatase inhibitors stop growth of tumor by preventing cancer cell to access estrogen. 3 aromatase inhibitors have been FDA approved. Taking aromatase inhibitor pill for 5 to 10 years improve disease-free survival and also lower the risk of developing secondary breast cancer. Another anti-estrogen agent is called fulvestrant. The fulvestrant was discovered from systemic medical chemistry. The structure is very similar to 17-beta-estradiol, but has additional group. So when fulvestrant binds to estrogen receptor, it will the estrogen receptor will be misfolded and processed by this garbage can called proteasome, and estrogen receptor is diminished. So fulvestrant is FDA approved in 2002. But because fulvestrant is not an oral drug, it requires intramuscular injection. Plus, it has unpleasant side effects. Fulvestrant is typically given to the metastatic breast cancer patient if they have developed resistance to other endocrine therapy. So another, for ER-positive breast cancer, another more latestly developed drug is called a CDK4/6 inhibitor. So tumor cell tends to have a rapid cell division. The cells after cell division, usually cell take a break in this G1 phase of cell cycle. CDK4/6 are the enzyme. They press the accelerator pedal, try to accelerate cell division. So the inhibitor for CDK6 inhibit the rapid cell division. So there are three CDK4/6 inhibitors approved by FDA. So when they are combined with hormone therapy, they greatly improve progression-free survival. However, these drugs are very expensive and the treatment resistance could also develop. So in a quick summary, for ER-positive breast cancer, for a premenopausal woman, tamoxifen is the first line of treatment. If the woman have ovary removed, aromatase inhibitor could also be prescribed. For postmenopausal women, aromatase inhibitor are the first line of treatment, followed by fulvestrant and tamoxifen. CDK4/6 inhibitor are given to all women at all age. So what I have told you about the estrogen, the receptor positive cancer, but not all the breast cancer express these hormone receptors. About 70% of breast cancer express either estrogen receptor or progesterone receptor. But 10 to 15% of breast cancer express a cell surface protein called HER2. Another 10 to 15% of breast cancer do not express any of these receptors. So they are called a triple negative breast cancer. So the treatment really vary based on the subtype of breast cancer. The HER2 is a cell surface growth receptor. It could send a signal to tell the cell to grow or divide. So for most of the normal cell, you only have a very few copy of HER2 on the cell surface. But some cancer cell could be coated with a lot of HER2. So they will send a more signal, cause cell to grow more rapidly. Herceptin is a monoclonal antibody. It's like a clamp to stop the HER2 from sending those growth signaling. Herceptin was approved by FDA to treat HER2 positive metastatic breast cancer in 1998. Treat of HER2 positive early stage breast cancer in 2006. So since HER2 has been used for breast cancer treatment, HER2 positive breast cancer have been dramatically, the deaths from HER2 positive breast cancer have dramatically decreased. So the HER2 positive breast cancer used to have the worst prognosis among all the subtype of breast cancer. So the triple negative breast cancer do not express this receptor, HER2, ER, or progesterone receptor. So the recurrence rate for triple negative breast cancer is very high as compared to the non-triple negative breast cancer. So therefore, there are still unmet clinical needs to identify new treatment for triple negative breast cancer. So I just talk about the treatment depends on cell type. The treatment strategy can be generally divided into a local treatment or systemic treatment. The local treatment include surgery and radiation. So the systemic treatment include chemotherapy, hormone therapy, targeted therapy, and immune therapy. So I will highlight a work by the McCardle faculty member. So this is a synthesis of 5-fluorouracil. This is one of the chemotherapy drug developed in McCardle in 1957, and it is still used in clinic. The 5-fluorouracil was synthesized by Charles Hadenberg, a solid tumor. So this is a picture from 1964. This is Charles Hadenberg along with other McCardle faculties. So breast cancer treatment depends on the type, and the stage also matter. The stage is defined by where is the breast cancer cells localized, and if they have spread to other tissues. So if cancer cells are confined to this ductal structure, so they are called stage 0. So those breast cancer are treated with surgery and hormone therapy. So as tumor grow, if they get out of this ductal structure, and also if a tumor grow in a bigger size, so they are called stage 1 to 3. So stage 3 are the breast cancer, the cell already spread to the surrounding lymph nodes. The stage 1 to 3 breast cancer are treated with surgery, radiation, hormone therapy, chemotherapy, and targeted therapy. Stage 4 breast cancer refer to metastatic breast cancer. So that means breast cancer cells have spread to brain, lung, liver, and bone. The 5-year survival rate for stage 4 breast cancer is only 30%. So the breast cancer, stage 4 breast cancer are treated with hormone, chemo, targeted, and immune therapy. But this is the breast cancer that take away most of the woman's life. So although we have a big success in handling breast cancer treatment, there are still some remaining challenges. For example, ER-positive breast cancer could become resistant to endocrine therapy. triple negative breast cancer still lack targeted therapy. And the triple negative breast cancer also don't respond well to immune therapy. And also we need to identify more biomarker to distinguish who will benefit from what treatment. So first, I will talk about this first problem, the ER-positive breast cancer become resistant to endocrine therapy. So with the advancement of the next generation sequencing, it is now possible to sequence just a few cells in the metastatic site. So these results are very surprising because it's found that the ER protein itself is mutated in this ligand binding domain. So this is where estrogen bind. So this mutation causes the receptor to be in a constitutive active conformation, as if the receptor already binds to the ligand. So tumors expressing this mutant estrogen receptor are more resistant to the existing endocrine therapy. We figured that maybe degrading estrogen receptor will be a good method to treat this kind of breast cancer. My lab discovered dystoinolacin G, which is a natural compound that could promote ER degradation. So dystoinolacin G, I call it DIPG in short, was originally isolated from stem barks of tropical plants. My former graduate student, Zibo Zhao, showed treatment with DIPG could lead to the ER degradation in breast cancer cells. So Christine, another graduate student, showed DIPG treatment could also promote degradation of the mutant ER alpha. So these results suggest that DIPG may be used to develop a novel therapy. In collaboration with Dr. Wei Tan in the School of Pharmacy, we have a rationally designed DIPG analog, which with improved efficacy to degrade ER alpha. A US patent has been awarded to our group. And now the the development of this DIPG compound into a drug is under development. And this study is supported by Department of Defense Breast Cancer Program. So the other challenge I will talk about lack of therapeutic targets for treatment of triple negative breast cancer and the lack of response of triple negative breast cancer to immune therapy. So if our immune system is fully functional, the immune cell, so here I will mostly refer to as T cells, will be able to recognize the tumor cells and eliminate them. This is because the tumor cell has expressed this protein called the MHC, so they can present a tumor-specific peptide derived from a tumor-specific protein on the cell surface that can be recognized by these T cells. And so this leads to a T cell killing of the tumor cells. So however, as tumor progress, the tumor cell tends to express the PD-L1, which interact with PD-1 in the T cells. So this is a friendly interaction. It's like they are shaking hands. Instead, the tumor cell hide their MHC molecule. The current immune checkpoint inhibitor is to use either anti-PD-1 or anti-PD-L1 antibody to disrupt these shaking hands. However, if the tumor cell do not express MHC protein to present tumor-specific antigen, the immune cell can still not recognize tumor cell to kill them. So my lab discovered that a protein we have studied for decades called CARM1 is highly expressed in triple negative breast cancer cells. And this protein modulates the interaction between T cell and the cancer cells. We find interestingly, the CARM1 is also expressed in the T cells. We find if we use a small molecule inhibitor to inhibit the CARM1, the tumor cell will express the MHC and presented this tumor-specific antigen. For the T cells, on the other hand, it's being activated. So their cells killing activity is dramatically increased. So this study established that CAM1 is a good target for improving the efficacy of immune therapy. Because the CAM1 inhibitor we used in our animal experiment was synthesized by GlaxoSmithKline, the GSK company, but GSK company has stopped developing CAM1 inhibitor in clinic. So we decided to design a new molecule that could lead to CAM1 degradation. So this is again with we collaborated with Dr. Wei Xu group. So here we design a small molecule. The one end binds to CAM1 protein. The other end binds to a component in this garbage disposal machinery. So in the middle is a linker. So when this molecule binds to both CAM1 and this E3 ubiquitin ligase, it will bring CAM1 into close proximity to this garbage disposal, and CAM1 gets decomposed. So this technology is called proteolysis targeting chimeric, also called PROTAC. So Megan has some data that seems very promising, showing that if she incubated this CAM1 PROTAC with the cells, we can observe like after 2 hours, CAM1 protein start to be degraded. And this decrease of CAM1 level sustained as we treated with this drug for a long time. So in this century, breast cancer diagnosis become molecular, and the treatment moved to a personalized treatment. So what do we mean about molecular characterization of breast cancer? Our human genome expresses about 20,000 protein coding genes. So if we profile this gene expression of breast tumors, so we can see they fall into these clusters. So here is if these genes are heavily expressed, but they don't express other genes, they're called luminal subtype A, and they are also luminal subtype B. HER2 is the gene name for HER2, so this is equivalent to HER2 positive cancer. The basal subtype is largely overlap with triple negative breast cancer. This is normal breast-like. So the advantage of doing this molecular characterization of breast cancer is that we can better predict patient survival. So for example, you can see luminal A tumor tends to have a much better survival than luminal B, followed by triple negative and ERB2, the HER2 positive. You may be surprised that the HER2 positive cancer has the worst survival. So this is because the study was published in 2003, before Herceptin was widely used for treatment of HER2 positive cancer. So now with Herceptin and a similar drug approved by FDA, Herceptin positive cancer now have a better survival than triple negative breast cancer. So a successful example of molecular profiling is also development of the TROP2 targeted antibody targeted therapy. So this is a new therapeutic agent for treating triple negative breast cancer. So the TROP2 is another cell surface protein that is highly expressed in triple negative breast cancer. A humanized antibody for TROP2 was developed. Importantly, this antibody could conjugate a toxin called SN38. So when the antibody interacts with TROP2, the antibody is engulfed into the cell, and the toxin is released to kill the TROP2 positive cells. And importantly, this toxin can also penetrate into TROP2 negative cell in the surrounding environment to kill those TROP2 negative cells. this is called a bystander killing. So this TROP2 targeted antibody conjugate was approved in April 2021 as the first antibody drug conjugate for triple-negative breast cancer. So this is used for treatment of triple-negative breast cancer patients who do not respond to chemotherapy. So a lot of time friends ask me, am I at a risk of developing breast cancer? So I will say around 10% of breast cancer are caused by the inheritance of damaged gene. So you probably heard about the BRCA1/2. So those genes were discovered in 1990s. The mutation on these genes are associated with increased breast cancer risk. In the last few years, additional genes have been identified. So now around 10 genes whose mutations are associated with increased breast cancer risk. So if you have inherited a bad gene, the risk of developing breast cancer could be as high as 80%, depending on the gene and the family history. So if you have a multiple family member diagnosed with breast cancer, a genetic test is advised. So the genetic test is simply a blood test covered by insurance. But 90% of breast cancer do not have a strong genetic association. So the breast cancer developed as we are getting older because more mutation could be accumulated. All women with dense breast have a higher risk of breast cancer because this provides a better environment for breast cancer cells. And a woman with a long menstrual period, so like early menstrual cycle and late menopause, they have a extended exposure to estrogen, so that tends to have an increased risk of breast cancer. So now with the molecular characterization of breast cancer, it is possible to design precision medicine for treating breast cancer. So circulating tumor cells are used for early breast cancer detection. The genetic makeup are used to tailor the treatment for individual patients. So for example, the HER2 positive cancer are treated with Herceptin. The BRCA mutant carrier are treated with PARP inhibitor. We can also follow the genetic change as the patient had gone through the treatment. So this is made possible through the next generation sequencing. The results will be interpreted by physician scientists in molecular tumor board. And lastly, we can also derive a patient organoid from the patient tumor and test different drug and drug combination. So this will likely give rise to a best treatment to the patient. So at UW Comprehensive Center, Dr. Barkhard and Dustin Deming are the two physician scientists that spearhead precision medicine molecular tumor board. So they analyze the tumor genotype and molecular abnormality in this tumor and make a recommendation for patient-specific treatment. I will briefly discuss one work related to this development of a patient tumor organoid and how we use this for drug screen. So we want to know if diptoinnonasen G, the compound I discussed earlier, has potential to kill tumors. So Christine cultured the 3D organoid derived from a ER-positive breast cancer patient. So then she treated this organoid with a control compound, DMSO, diptoinnonasen G, and a positive control, fulvestrant. So the results show that DIPG and fulvestrant are both effective in killing this tumor organoid. So this provides a strong evidence that DIPG may be developed as a new type of drug for treating ER-positive tumor expressing mutant ER. So the other project in my lab I will not discuss today deal with the problem such as ER positive tumor lost ER expression. How should we treat those patients? Endocrine resistance caused by tumor expressing both ER and HER2. And we also work on identifying new biomarker for predicting which patient population could benefit from a new treatment. So in summary, breast cancer has the longest treatment history in the medical oncology. And we know a great deal of breast cancer biology. So breast cancer is among the most curable cancer. The treatment evolved over the past 2 centuries and become more targeted. Breast cancer treatment has moved to a molecular era of precision medicine, and the new targeted therapy and biomarker remain to be explored. So there are still some questions we need to resolve. So with that, I would like to thank my lab members, this younger generation scientists, very dedicated to breast cancer research. And we like to thank the funding agency for providing the financial support for our research. And we also want to thank Promega for providing the reagents, and of course the Carbon Cancer Center for supporting some pilot projects. So our project is not possible without collaboration with various groups at UW-Madison. Dr. Peng Liu, Marian A, and Hui Ding are the bioinformaticians, so they analyze big data for us. Mark Burkhardt, Kevin Winsinski, Josh Lang, and Lee Wilkie, they are oncologists dealing with the patient treatment. Wei Ping Tang and Lin Jin are the faculty member in School of Pharmacy. They help us with medical chemistry as well as proteomics needs. We also want to especially thank patient advocates, Chris Miller, Katrina Phillip. They joined my lab meeting before the pandemic starts. That provide a specific perspective from the patient point of view. So with that, I'm happy to take any questions.
Thank you so much. And it clearly takes a village, right, to accomplish everything that has happened over the past several decades. Very impressive research. It's like decoding a puzzle, huh? It really is fascinating. Thank you so much. Hello, everybody. Fran Paleo Moyer, Badger Talks producer. We're talking to Dr. Wei Xu today about breast cancer history and biology, particularly at the University of Wisconsin and in Wisconsin. Feel free to post any questions in the chat that you might have. A couple of questions. Well, first of all, I want to acknowledge your thoughtful acknowledgement of the history and of all of the people who really have played such an important role in your department and with all of the findings. And then also, of course, the work of the graduate students, including Megan, who introduced you today. I love that you highlighted her work, so thank you so much. So what's really become evident as we've gone through this series of talks with the McArdle Lab faculty is that cancer treatment has now become very specific to each individual. Can you tell me, and that's clear through all the talks we're hearing that, can you tell us how you think that will shape treatment in 25, 30 years? I mean, I can envision everybody having sort of like a chip, and we know specifically what genes everybody, you know, in my little science fiction brain, of course, that we can just kind of predict that for everybody. But tell me what you think could be the future.
Mm-hmm. Yeah, certainly. Yeah, this is an important question. I think our ultimate goal is to eliminate cancer, to cure the cancer. So early breast cancer detection is a key. So like a mammogram is now very widely used for early breast cancer detection. Circulating tumor cells, tumor DNA are also used for early detection of breast cancer. So if any cancer could be detected as an early stage, so there is a way to get the patient cured, because you can just surgery remove the cancer so that they haven't spread to other places. So yeah, so for the early detection of breast cancer, yeah, using this newer generation sequencing technology, you can also learn the feature of this cancer. So what kind of a mutation are in these tumor cells? What kind of genes are expressed in this tumor cell? So all those information will inform the clinicians to make a recommendation of what treatment will be useful for individual patient. So that is in general called the precision medicine or personalized treatment.
Is it a viable idea that genes could change in the future if we continue to repress or suppress cancer from happening? Or?
Yeah, that is a very good question. So that is called resistance to a treatment. So a lot of time we find the patient, the tumors are responsive to certain treatment, but tumor evolve. So they try to get away from that drug. So there are certain clones. Yeah, you can think this is a tumor cell, but there are just a few chunks, they develop a new mutation to escape the original treatment. So therefore, we have to keep using this next generation sequencing to follow them, see what they come up to, what kind of a new mutation they develop. So then we know maybe there are certain tool already become available, or something we can design a new treatment to treat those resistant tumors.
Thank you. And how do you account for the parts of the world where there's very low incidence of breast cancer based on your initial map that you had that you showed of the world, and that there are parts where it's not as common? Those people clearly have a different gene, is that right? And that is why, or how does that work?
Yeah, that's a very complex questions. So there are multiple factors. Genetics certainly contributes to it, but there are also other factors. This is a question related to breast cancer prevention. So maybe if you exercise a lot and you keep watching your weight, once you become more healthy, so the likelihood of getting breast cancer is also low, because obesity is another risk factor associated with breast cancer. So for the woman who have gave birth to a children, more breastfeeding time is also protective for women to develop breast cancer. I think in a lot of Western country, yeah, because we are so busy, we don't have time for breastfeeding for a very long time. So maybe that contributed to a high risk of breast cancer. So it's very complex.
I see that. We have a question posted by Jill Schneider. She's asking, should postmenopausal women with dense breast tissue be screened for breast cancer with methods other than screening mammograms?
Mammogram in general is very sensitive. So that is recommended if for the woman with a dense breast. But if you also have a family history with breast cancer, so certainly you need a genetic testing. Those are the DNA tests to screen for the genes. But in general, mammogram is recommended for early breast cancer screening.
You had also indicated on one of your slides the premenopausal and postmenopausal factors. And I'm wondering, you mentioned obesity is another factor. Like, what are all of the factors that are considered, and would age also? I know, certainly people I know in their 80s and beyond are being told maybe don't do screenings as much because maybe it progresses very slowly at that age. How does age factor in?
Yeah, the age is an important factor. In general, for the majority of the cancer, as we age, we pick up more mutations. So we are at a high risk of developing cancer, including breast cancer. But for breast cancer, it's very specific. There are certain breast cancer associated with pregnancy. So this happens in the premenopausal woman. So the pregnancy could also stimulate breast cancer for some women. For premenopausal women, taking taking contraceptives containing estrogen and progesterone could also increase their risk of breast cancer. So for postmenopausal women, so there has been a large clinical trial showing postmenopausal women taking the hormone replacement therapy. So those also contain the hormones, the estrogen, progesterone. So those could also increase the risk of breast cancer. So therefore, now the hormone replacement therapy is not commonly, it's not recommended to every woman. So you want to discuss that with your doctor if you need it for treating postmenopausal symptoms.
Okay, thank you for that. And one last question. How does this research get shared with doctors? Obviously, you're affiliated with the UW hospital and being a teaching hospital, but around the state in general, how does information from the research that you're doing get shared?
Yeah, so I actually interact a lot with the physician scientists like Mark Barkard, Kevin Winsinski. So we have a lot of research meetings going on. So Kerry has been a co-author on multiple papers my lab published. So yeah, we share information through a frequent in-person meeting. Of course, it's changed to Zoom during pandemic, but we always discuss with them and see what kind of a clinical trial are going on at UW Comprehensive Cancer Center, see if we can test certain hypotheses we have in the lab using the patient sample that collect from the clinical trial. So Kerry Winsinski, I know, is also part of the Big Ten consortium. So they also share information with other universities through that consortium. So yeah, a lot of information we gathered can be quickly shared with the physician scientists, and we also could reshape our research based on the feedback we get from them.
Well, thank you so much for sharing your important work. We all, as I've said with the other talks, know people who've been impacted, and I'm sure have been impacted by your research directly. So thank you so much today for sharing this information with us.
Thank you very much.
Great. Well, that concludes our incredible series of the research happening at the McCardle Lab for Cancer Research. I'd like to give another special thank you to Emeritus Faculty Richard Burgess, who helped put this series together and really was an integral part of it happening. So these talks will remain out on the Facebook, Badger Talks Facebook and YouTube pages. So please share them with people who you know might be interested in this information. We're going to kick off a new series starting next Tuesday, October 4th at noon. We're going to be working with the School of Human Ecology, and we're going to be talking with Dr. Sarah Ann Carter, who's the director of the Center for Design and Material Culture at UW. And she's going to be talking about the 25-year history of material culture at UW and giving us a peek at one of the new exhibits there. Please check out badgertalks.wisc.edu. We just posted a series of upcoming talks coming up. You can also sign up for our email there on that website. Consider a donation to Badger Talks. We are funded by a grant. Also sign up for our email list so you're the first to know about upcoming talks. And you can also search the roster of over 400 UW faculty and staff who have generously signed up to donate their time to talk in communities like yours around the state about issues that are important to you. Thanks for tuning in, and we'll look forward to seeing you next week.